Hello students, welcome back. Welcome back to my biology online classes. In my previous lectures, I have already discussed about the detailed structure of DNA, the famous Watson and Crick model and their descriptions, and DNA polynucleotide structure, detailed nucleotide structure, etc. Today, I am going to discuss with you about the types of DNA. How many types of DNA? First of all, the Watson and Crick model of DNA we have already studied. That one is B-DNA. That one is right-handed DNA. There are 5 types of DNA. Okay, A, B, C, D, and Z DNA. This one is Watson and Crick model DNA, which I have already discussed. Right-handed DNA and the most abundant, most common type of DNA. Now see the point of difference. Point number 1. Number of term. Number of base pair per helical turn. Okay. What is helical turn? I have already discussed in my previous lecture. If you have some confusion, please consult with that lecture. A DNA, 11 base pair per turn is present. In B DNA, 10 base pair per turn is present. And in C DNA, 9.33 base pair per turn. And in case of Z DNA, it is 12 per turn. Next point, coiling. Here, A-DNA, right-handed coiling, a clockwise coiling, and right-handed coiling. Here only in case of Z-DNA, left-handed or anti-clockwise coiling is present. So, this is major difference. This is main difference between these 3 types of DNA. As all of you know that, as all of you can see that Z DNA, it is only this one is left hand diameter. Diameter of B DNA is 2 nanometer. Diameter means it is also discussed in my previous lecture. Please consult what is diameter of DNA. 2.6. Of Z DNA, it is 1.8 nanometer. Pitch length. In B DNA, it is 3.4 nanometer. And what is pitch length? To know this point, please consult my previous lecture. 2.86 nanometer pitch length. In Z DNA, 44 nanometer pitch length is. Let's discuss something more about A DNA. Observed under low humidity or dehydrating condition is shorter and wider than B DNA. Point number 3, DNA adopts this double helical form and DNA duplexes are formed only in A form and form RNA, DNA duplex. But B form DNA RNA duplex also being in localized single-strand dinucleotide contexts. RNA can also adopt the B form without pairing to DNA. DNA has a deep, narrow major groove. Deep, narrow, major groove, which does not make it easily accessible to protein. Wide, shallow, minor groove is present here. Its favored condition is low water condition. ADNA's base pair are tilted relative to the helix axis and displaced from the axis. Tilted. Next point, it has several biological functions. So it's all about ADNA, BDNA. The detail of BDNA I have already discussed as Watson and Crick model of DNA. This is the most common form of DNA. Most common form of DNA. Next point, more narrow elongated axis than ADNA. Its wide major groove makes highly accessible to proteins. BDNA is found in high water condition. High water conditions. BDNA base pairs are nearly perpendicular to the axis. It's all about BDNA. And come to the last and most important form and also exceptional form, that one is zDNA. zDNA, it was discovered by Andres Wang and Alexander Rick already discussed. This one is left-handed double helix and it is found in high salt concentration. It is formed at high salt concentrations. Point number 3. This one is more narrow, more elongated than A and B DNA and it have narrow minor groove. And the major groove of Z DNA is not really a groove, presence of narrow minor groove. So it's all about different types of DNA.